Mr. Chairman, ladies and gentlemen, on behalf of Bayer, I shall give you a brief glimpse into Bayer's oncology pipeline, and I will at the end focus on two particular projects in colorectal cancer. Drug discovery at Bayer is located at four major sites, as you can see here, in Berlin, in the Wuppertal-Cologne area in Germany, in Berkeley, San Francisco, and in Beijing. Oncology drug discovery is mainly concentrated in Berlin, while biologic research in oncology is done in the research center in Wuppertal. Also, the Center for Innovative Drug Discovery in Beijing is active in oncology drug discovery activities. Bayer invested 2 billion euro last year for research and development. That represents about 11% of total net sales. The total number of employees in research and development in 2013 was 7,800. 2,700 of them are actually working in global drug discovery. Bayer relies on a vast network of collaboration, as you can see here on this slide, both for drug discovery of particular research assets and or for using technologies, as well as for drug development and distribution. To name just a few, we entered into an extensive collaboration with OncoMed in terms of anti-cancer stem cell programs, in particular to develop various substances targeting the Wnt pathway, for instance, ventictumab, a pan-frizzled monoclonal antibody. I will come to this shortly later. Or OMP55F28, a fusion protein that consists of a portion of the frizzled 8 receptor from the Wnt pathway fused to the Fc domain of a human IgG1. So it inhibits the Wnt pathway by binding to the Wnt ligand. Another collaboration is that with Prometheus. The collaboration partners Prometheus' proprietary oncology diagnostic platform, in particular in terms of mutational analyses, with Bayer's broad oncology pipeline in an effort to stratify patients to appropriate drug candidates and to potentially accelerate the development of novel oncology therapeutic products. The license agreement with Celera provides Bayer access to 5 cancer-related targets for therapeutic development and in vivo diagnostic imaging. These therapeutic targets are overexpressed on the surface of several different tumor cell types and were identified by using Celera's proteomics discovery platform. In September last year, a strategic alliance between Bayer and the Broad Institute of Harvard University and the Massachusetts Institute of Technology was announced in the area of oncogenomics and drug discovery. The goal of this collaboration is to jointly discover and develop therapeutic agents that selectively target cancer genome alterations, and this collaboration is aimed to cover the next 5 years. Last but not least, there exists a strategic alliance between Bayer and the German Cancer Research Center, our today's host. The last press release coming out of this alliance dates from April last year, announcing the opening of a joint immunotherapy-focused research laboratory within the National Center for Tumor Diseases with a joint investment of 3 million euro. In general, the partnership supports the strategic goals of both partners and contributes to the development of innovative therapeutic options for patient care. The alliance between Bayer and the DKFZ is an ideal partnership as strategies of both fit and unites complementary expertise. Joint projects have a strong impact on our early development pipeline and an expansion 2 clinical projects is planned and has already started. Bayer's research and development activities focus on 4 therapeutic areas: cardiovascular diseases, oncology, women's healthcare, and anti-infectives, whereas oncology represents 40% of clinical pipeline projects marked in red here on this slide. There are currently 14 projects in phase 1, 14 in phase 2, and 16 in phase 3 stage of clinical development. This is a more detailed view of the oncology pipeline, showing both substances in preclinical and clinical development. In general, there is a whole raft of molecules. I'd like to mention here also just a few. A tumor necrosis factor receptor antibody whose regulation and ligand play a role in breast cancer cell invasion. A BOB1 inhibitor playing a role in cell cycle inhibition. A BRD4 inhibitor for chromatin modification, an allosteric ACT1/2 inhibitor, a fibroblast growth factor receptor 2 antibody inhibiting both angio and stomatogenesis, an oral and an IV PI3K inhibitor. The IV one, copanlisib, is in development for non-Hodgkin's lymphoma currently. A mesothelin antibody drug conjugate, one of our focuses in biologic research, coming from the collaboration with Seattle Genomics, a CDK inhibitor, and others. Also to mention that we do maintain an active life cycle development for our already approved products, sorafenib, regorafenib, and radium-223, to broaden their spectrum of indications. Here, again, you see which areas of tumor genesis are addressed by the various substances. These comprise, but are not limited to, cell cycle and apoptosis, the RAS, PI3K, and the APC pathway, chromatin modification, as well as transcriptional regulation. Now I'd like to focus on 2 molecules for which a combination treatment seems promising also in colorectal cancer. The first of the 2 is refametinib, an oral MEK inhibitor. As you know, the RAS-RAF-MEK-ERG pathway is one of the main pathways deregulated in cancer. It controls cellular proliferation, differentiation, apoptosis, and migration. Refametinib is an oral potent non-ATP competitive inhibitor targeting MEK1 and MEK2, the central switch in the RAS signal transduction cascade. Preclinical data have provided a robust rationale to use refametinib in combination with other pathway inhibitors such as multikinase, PI3K, ACT, and mTOR inhibitors. In vitro anti-tumor activity has been shown in preclinical tumor models of hepatocellular carcinoma, pancreatic cancer, melanoma, colorectal cancer, and NSCLC. The phase 1 study has been conducted and examined the recommended phase 2 dose, which was determined with 50 milligram twice per day. Refametinib binds in a unique allosteric pocket adjacent to the ATP binding site. This allosteric site is found only in MEK1 and MEK2 and explains the exquisite selectivity of this compound. Here, the anti-proliferative activity of refametinib in comparison to 2 other MEK inhibitors in various cell lines are shown. You see a strong anti-proliferative activity of refametinib in HCC in the top section, pancreatic cancer, colorectal cancer, and non-small cell lung cancer that carry an activation of the RAS-RAF-MEK-ERK pathway. Synergy with EGFR inhibitors it was observed in several KRAS mutant cell lines. mTOR inhibitors appear to synergize with refamitinib in PI3KCA mutant lines, and synergy with gemcitabine, the last but one column, occurs in several cell lines. However, there are also strong hints of a good synergy with our multi-kinase inhibitor regorafenib, which is already approved for metastatic colorectal cancer and is awaiting approval by the EMA for gastrointestinal stromal tumors shortly. Regorafenib is a potent oral multi-kinase inhibitor with a distinct profile targeting angiogenic and oncogenic receptor tyrosine kinases. Regorafenib effectively inhibits tumor cell proliferation as well as tumor-induced angiogenesis. The preclinical rationale to combine refamitinib and regorafenib comes, among others, from these experiments using the murine syngenic MC38 colorectal cancer liver metastasis model. The combination of both substances, here depicted in the pink curve, more than doubled median overall survival in this animal CRC model. However, there are other entities for which in vivo rationale exists. Here, for instance, in a breast cancer xenograft model, the combination of the two drugs is significantly better both in the primary tumor and in the percentage of animals with lymph node metastasis. And finally, although mixed, there's also evidence supporting the combination of the two drugs in patient-derived gastric cancer xenografts. Although in most cases only minor benefit of the combination is seen in comparison with the respective monotherapy, the combination appears to be, at least in some tumor metastasis, to be better. Based on the information just presented to you, we are currently setting up a clinical study in which the combination of refamitinib and regorafenib will be explored. The drug-drug interaction potential is considered to be low. However, there might be considerable potential for overlapping toxicity. Hence, a formal phase 1 dose escalation study is necessary to be conducted. In the finally to be conducted phase 2 part of the study, 3 cohorts of patients will be enrolled, those with colorectal cancer in 3rd or beyond line, both bearing a RAS mutation or a RAS wild type in their tumor, NSCLC patients treated in 2nd or beyond line, also with KRAS wild type and KRAS mutant cancers, and breast cancer patients after anthracycline and taxane treatment. Another area of research, which I alluded to earlier, is the manipulation of cancer stem cells in a way that relapses which are generated by the permanent existence of these cells are prevented. This can be done by inducing a differentiation signal in these cancer stem cells in order for them to become mature differentiated cells. One of those potential cancer stem cell inhibitors is vantictumab, coming from the collaboration with OncoMed. Vantictumab, also known as OMP18R5, is a fully human IgG2 antibody targeting multiple frizzled receptors. By means of this mode of action, it acts as antagonist of the Wnt beta-catenin signaling pathway, which is important in cancer stem cells. The phase 1 dose escalation study with vantictumab in subjects with solid tumors, including colorectal cancer, has reported first results. In terms of dose escalation, 6 out of 7 planned dose groups have completed enrollment. The last group, the 10 milligram per kilogram every 3 weeks group, is continuing enrollment at the moment. There were prolonged responses seen in 3 neuroendocrine tumor patients and a minor response in a pancreatic NET patient, which may represent single agent activity. In terms of biomarkers, which is more interesting in this study, it was found that vantictumab inhibited the Wnt target, stem cell, and epithelial mesenchymal transition genes and increased the expression of differentiation genes. Rationale for combination development could be this finding in a preclinical breast cancer model. The combination of vantictumab and Taxol, the yellow curve, is very active in this tumor model in comparison with the curves showing the combination of Taxol with other cancer stem cell targeting substances, here with an anti-Notch and an anti-DLL4 agent. Tests with other combination partners in CRC models are ongoing. By sharing these results with you, I'd like to end and thank you for your attention.
Thanks, Mr. Strauss, for your excellent presentation. Questions? One question from here, perhaps. Can you say one sentence more about your collaboration with the German Cancer Research Institute?
Well, this collaboration exists for, I think, now 5 years, and the interactions on the preclinical level, I think, are very strong. There is room for improvement from my perspective in terms of clinical studies.
I have a question. Obviously, aspirin is very important in this field with regards to prevention. Are there any other activities with specific regards to prevention in the upcoming portfolio at this time?
Not to my knowledge, no, not at the moment, in a way similar to aspirin. So I doubt that we will engage in other patent-free things, and I can't see anything on the horizon which would act as prophylaxis of any type of cancer at the moment.
Okay, thank you.